actually look at the cascade of events here. So this is kind of what we began speaking about in the last unit. We talked about metabotropic receptors. We talked about how they're coupled to this G protein. And we began sort of outlining what this process looks like. When we got to this part of the process, we were very vague, okay? And I told you all that we would pick up and fill in the details when we get to the actual signal transduction mechanism, okay? So this is a metabotropic receptor. Now, this is specifically the mechanism for alpha-1 receptors only. Alpha-1 receptors are different in this way. They use an entirely different mechanism. They use an entirely different second messenger, okay? So we'll talk about alpha-1 first. So this should be marked alpha-1, so you can mark that in your book, just so that you're clear that this is only possible at an alpha-1 receptor, okay? Norepinephrine or epinephrine, now which neurotransmitter does an alpha-1 receptor prefer? What does it have a higher affinity for? Epinephrine or norepinephrine? Alpha-1 receptors prefer norepinephrine. Okay, so norepinephrine would have a higher affinity here. Okay, so let's say norepinephrine binds, it's coupled to this G protein. Let's walk through the sequence of events here. Now, this G protein will undergo a conformational change upon the binding of that neurotransmitter to this receptor, such that the alpha subunit will dissociate off of this protein and it will make its way down the membrane and bind to another enzyme that is in the membrane. This enzyme is called phospholipase C, okay? Now, if you look at the terminology of this enzyme, a lipase is an enzyme that cleaves or cuts another molecule, okay? Lipase, when it ends in ASE, it's cleaving or it's cutting. So that's going to tell us a little bit about the job of phospholipase C, okay? So we have this subunit activating phospholipase C, and phospholipase C is going to cut or cleave this molecule called PIP2. Now, you don't have to know what PIP2 stands for at this point, but it's phosphatidylinositol phosphate, very long word to say. So PIP2 then gets cleaved into two other components, two other molecules. The first one is IP3, okay, IP3. The second one is DAG, okay? So DAG and IP3 together are the two byproducts that came about from cleaving PIP2, all right? Now, these are our second messengers, specifically DAG. DAG is going to enter the cell, okay? It's going to turn on enzymes, protein kinase C. Protein kinase, we talked about this early in the semester. If we see kinase in the name, it is going to be phosphorylating or adding a phosphate group to another molecule. So protein kinase, once it's activated, can phosphorylate some protein in the cell. Now, this is where things get a little bit general, a little bit vague, because depending on what cell we're in, we will bring about a different response, okay? But the big take-home picture here is the second messengers here are DAG and IP3, specifically DAG, okay? Because DAG is going to activate protein kinase C. Protein kinase C will phosphorylate some other protein in the cell, and that will bring about a cellular response. Now, protein kinase C and DAG can also elicit calcium release. So this can be a part of the response as well. If we elicit calcium being released from the endoplasmic reticulum, this is the organelle that stores calcium in a cell, and that release can help to bring about or facilitate cellular response, okay? So I'll go over this one more time. We have norepinephrine, more than epinephrine, binding to an alpha-1 receptor. This receptor is coupled to a G protein. It undergoes a conformational shift or a conformational change. Now this GS here is telling us that this is a G stimulatory protein, meaning that this alpha subunit will stimulate or activate or turn on phospholipase C. Once we turn on phospholipase C, it will cleave PIP2 into two smaller molecules, IP3 and DAG. These are our second messengers. These are the guys that are going to enter the cell and go and do something to bring about cellular response, okay? DAG enters the cell. turns on protein kinase C, which is another enzyme floating around in the cell, and protein kinase C will phosphorylate some other protein depending on where the cell is, and that will bring about our cellular response, okay? Likewise, DAG can also facilitate calcium release from the endoplasmic reticulum, which will also help to facilitate a response. Okay, so that was alpha 1, and that's the unique mechanism only seen for alpha 1, okay? Alpha 2 and all beta receptors use a different mechanism entirely, okay? They use a different second messenger, they use a different enzyme. Now, this image is usually very confusing for students, and that's because of the sort of schematic nature that this is being shown. So alpha 2 is on this side, whereas all beta receptors are on this side, okay? So it's two different processes happening here. So let's talk about alpha 2 receptors first. Now, alpha 2 receptors will bind, again, norepinephrine. This is coupled to a G protein, but this is a G inhibitory protein, okay? This subscript I means that this protein is going to inhibit its enzyme. So when the subunit dissociates off, it's going to turn off or negate the enzyme that is downstream in the membrane, okay? And so this enzyme happens to be adenylate cyclase. And so binding of norepinephrine to an alpha 2 receptor turns off adenylate cyclase, okay? This is an inhibitory second messenger pathway. This will turn this cascade off, okay? On the other side, on this side, this is the mechanism for all beta receptors. Now, beta 1, beta 2, beta 3, they all use this mechanism. So make sure you make a note here that this can be beta 1, this can be beta 2, this can be beta 3. These beta receptors bind, again, epinephrine. Now, this is where things get a bit tricky. There are varying affinities here. So you can get a question that asks you, if it's a beta 2 receptor, which neurotransmitter it's going to bind more likely? If it's a beta 2 receptor, it's going to bind epinephrine with a greater affinity, okay? So it's going to bind epinephrine. There will be a conformational change here again, and this is a stimulatory protein, so that will turn on or activate adenylate cyclase. Adenylate cyclase will synthesize cyclic AMP. So cyclic AMP is acting like DAG in this scenario. It's our second messenger. It's going to enter the cell. It's going to turn on protein kinase. That's going to phosphorylate some type of protein, and that will bring about a cellular response, okay? Okay, so I'm actually going to walk through it here now just to repeat this. We have an alpha 2 receptor, which is inhibitory. It's going to bind to this, norepinephrine will bind to this receptor. That will bring about a conformational change, and we will turn off or inhibit adenylate cyclase, okay? So we don't have second messenger entering the cell, and that is actually a part of our response. We'll talk about the response that's brought about in a minute. Alternatively, on the other side, we have norepinephrine or epinephrine binding to a beta receptor, and that will turn on, because this is a stimulatory protein, adenylate cyclase, which will create cyclic AMP, phosphorylate or turn on protein kinase, and protein kinase will phosphorylate some protein in the cell that will bring about a cellular response, okay? Now, just like I mentioned earlier, we're gonna sort of fill in the gaps of what happens here when we move on in the semester. When we talk about the cardiac muscle, when we talk about the skeletal muscle, we're gonna fill in the gaps of exactly what protein is turned on, how it brings about muscle contraction or cardiovascular function when we go on. So the details that I really want you to focus on with these, at this point, is what neurotransmitter binds to what receptor, right? What is the affinity of it, the neurotransmitter to that receptor, what the second messenger is, and whether or not it's activating or inhibiting its second messenger, okay? All right. Now, this table is essentially just that. This is sort of a good study table, a good summary table. This is all I want you to focus on. Beyond this, it becomes very complicated and beyond the scope of this class. So you need to know the receptor type. You need to know the tissue that it's found on, right? So for example, beta 3 is only seen on adipose tissue. You need to know the relative affinities. So what neurotransmitter does this receptor prefer? Okay, you need to know the signal transduction mechanism. And when I say signal transduction mechanism, this basically means what is the second messenger that's being used, and what effect does the receptor have on the second messenger? So for example, the second messenger could be the same, but one, for example, alpha-2 inhibits its second messenger, the other is activated. And then lastly, you need to know the effect on the tissue. So again, the effect on the second messenger differs from the effect on the tissue, okay? Don't get those confused. All right, this is another summary table which sort of highlights some common drugs, some common medications that use these receptors that we use to treat some common symptoms. A good example is isoproterenol. So isoproterenol is a beta-1, beta-2 agonist, meaning that when we give patients isoproterenol, it mimics the response of norepinephrine binding to a beta-1 or beta-2 receptor. And that is good because if we have norepinephrine binding to beta-1 and beta-2 receptors, we get bronchodilation. So remember we said that that inhibitory effect on the tissue is to dilate the tissue, dilate that smooth muscle. And that's what we want in asthmatics, right? For asthma patients, to dilate the bronchioles. So we can use this receptor neurotransmitter relationship, and we can sort of hone in on that, capitalize on that, and make some medications that can mimic these responses, okay? To treat glaucoma and dry mouth, we use muscarinic agonists because the parasympathetic system increases our secretions, increases lacrimal secretions, increases tears, increases salivary secretions. And so we can, in patients with dry mouth or dry eyes, we can capitalize on that effect and mimic the parasympathetic response, which will be beneficial to those patients, okay?